So actually, I'm not going to talk about necessarily reproducibility in general, but actually a very specific part of this. And at least it is the most cost-effective way of making your lab work more reproducible and more visible to your colleagues in general. So I want to summarize, let's say, split my talk into three different things. One is just a summary of some of the problems that I find, and I think many others find, when it comes to reporting of lab work, methods, materials in science in general. Then I'd like to introduce one platform that may improve this and mitigate some of the problems that we have in science reporting. And finally, I would like to do a little FAQ, so before the general Q&A, but a little FAQ of things that very commonly come up when I talk to people about this, to head off some of the questions that may come up, but you are welcome to ask again those questions, and we can discuss in a bit more detail afterwards if you're interested. So to begin, what are the problems that many of us face when it comes to reporting of like materials and methods and protocols in lab work protocols, specifically in archaeological science or science in general? So actually, I would like to do sort of audience participation here. So please get your hands up on Zoom ready. So my first question is, who likes reading materials and methods sections in academic publications? Okay, that's 4 out of 58. Oh, okay, there you go. About 15 or so. Okay, you're a bit, oh, it's going to quiz me. Okay, fair enough. Maybe it's not going to go the way I was expecting, but anyway. Next question. Who enjoys writing the same materials and methods sections for each of your publications over and over and over again? So the same protocol, but over and over again. Okay, that's not so many exactly. Okay, that's better. More what I was expecting. And finally, last question is, while you're in the lab, so for the ancient DNA people and the paleoproteomics people, for example, you know, dressed up in your clean room suit, who actually follows the protocol based on the original PDF of the publication it was published in? Okay, very few people. That's basically what I expected. So the reason why, in my opinion, methods sections suck in most cases, at least from a practical point of view, is that they miss all the detail. 21st century science still relies on basically prose that was developed in the 1900s or so. And increasingly publishers are putting overly restrictive word count limits on us, which means that we have to remove a lot of the detail that we actually have to put into our increasingly complex experiments, increasingly complex protocols and analysis. And ultimately, the loss of this detail means that much of this is not reproducible based on this, let's say, source of truth that comes with our publications, which is the methods materials sections. For example, this is an example of what I did in my first publication, literally the entire lab work, which I spent 5-6 months developing, I summarized in about 2 lines. But you can see that with that information, those 2 lines, you're missing a lot of the critical information you need to know about how that data was generated and how then you need to necessarily analyze it. You may argue, oh, but James, you've cited the protocols that you've used there. Fair enough. But can you really trust that? You can never really know exactly what they did. Can you really trust when people rely on things like citation chains? So examples from ancient DNA publications here, I've anonymized it. Oh, this is common in many, 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 many. This is one particularly epic one I found, where because of the space limits, they basically cited, well, we use this publication. This publication points to the person's supervisor, who actually cited some of their previous lab back during their early PhD, who eventually goes down here. But what you can see is they're all saying modified, described by, modified, described by, modified, described by, but this is in 4 different labs. And in fact, this last publication was actually a methods testing paper where they developed different protocols. So there's no way of knowing actually necessarily which one they picked. So again, this is quite bad because you you don't know actually what the people did. You cannot reproduce their data. You've potentially lost critical information or tweaks or modifications that people made to generate their data. And in fact, which I love this because it came out like last month, a paper came out scientifically showing just how bad citation chains are for reproducible science in biology, neuroscience, and I think it was psychology or sometimes psychology, where they systematically went through hundreds or thousands of papers and tried to see how common are these citation chains and how easy is it to get the information you need for this. And what they found is, like, for example, in this one biology paper, of the, I think what this, 13 citations, they were never actually able to get to the original method sections of many of them because they always hit a paywall. In some cases, they had the wrong citation. In other cases, it pointed to a different protocol, which is not actually relevant to what they were doing here. So with all of this, this is really bad because you cannot get this critical information, you know, which polymerase you use, which machine you use to do this certain thing. So yeah, it's not good. Another frustrating thing, sort of again, more practically, is when you're writing these protocols up, you don't want to have to, let's say, in the lab, between two publications, you make a slight tweak to your protocol because, you know, maybe you replaced a machine, you have a new one, a new heating block, and it has different settings or whatever. You don't have to rewrite your entire protocol for a single line change for every single publication. That is annoying and a waste of time. And furthermore, if you're writing together in a group, in your lab, you're developing protocols, tweaking protocols together, you want to best do this in an efficient manner. You know, we do have Google Docs, Google Drive, and things like this, but often people fall back to things like Excel sheets. And if you're working on this together, you can get horrible messes like this, where you've got a common single file, which is your protocol, but then 20 people open it, they have lock files, they make changes, they don't save it properly. Someone goes and tries to clean this up, and they delete the entire directory. Your entire protocol record is gone. It's also really bad. So again, with this sort of archaic, arguably, forms of writing sort of things and things like Excel documents do not match the way we work today. And also, following protocols are annoying, except for the 4 people who put a hand up saying they actually took this into the lab. But I really strongly feel that this, what you see on the right, is not suitable way to follow a protocol in a lab. A protocol is a recipe. You want clear step-by-step instructions. You want clear notes and sort of markers indicating critical points and things like this. This is just horrible. You really have a high risk of making mistakes because, you know, you get confused which line you were on and you copy the wrong, you know, adapter or something like this. And writing all of this out is really time consuming. So let's say you have got your Excel sheet, you have for each publication, you have to go and rewrite that for every single publication you make, because also you have to make sure you don't get called out for plagiarism, even if it's your own work, but that can also happen in certain automated systems. So and having to rewrite this into, you know, from your Excel sheet to your prose, you also increase the risk of including, you know, typos, mistakes, which could be critical in many cases. And so ultimately, my argument is that we can save a lot of our time. So last poll, would you rather write something like this and read something like this? Or would you rather have to write just a 2-line thing and have a link to a protocol in a recipe format, which is something you can use both in the lab, that you can read properly when you're preparing your protocols and everything? I suspect most people would like to have this. Okay, well, it's another example of what I mean by this like step-by-step protocol thing, and this is a sneak peek of what you can see in a minute. This, I would argue, is much more readable than the stuff on the left. Who would agree with me? Put your thumbs up or hands up. Yeah, there you go. Sustained about 10, 15 people. It's not going down. Great. So now I would like to introduce Protocols.io, which is one possible option for adopting such a platform which allows us to publish protocols, show protocols in a 21st century science manner. So Protocols.io themselves, they call themselves a secure platform for developing and sharing reproducible methods. For me, the way I like to describe it, it's a cross between, a protocol publication platform for 21st century science, so not PDFs, not prose. It's also a platform a bit like Google Docs for lab protocols because you can collaborate with other people. And also, and most importantly for me, who's a zealot for reproducible research, is a GitHub for lab protocols, which provides versioning. So what I mean by this, as you saw in the sneak peek a few slides ago, is that you have this step-by-step layout, which is nice. You normally get this in traditional, say, protocol journals. If you publish the protocol, it's a protocol. But you have interactivity in this. So you can have collapsible notes. So if you're getting a new student in, right, they need to have all the details, specific guidance, all these like tips and tricks they may need to get this to run. You, if you're an advanced lab tech, you've been doing this for 20 years, you don't necessarily need all these notes, so you can collapse them. You can have special like buttons and icons to indicate critical bits of information within the protocol. You can have tables, things like this. Also, because this is on a HTML, so a platform website, there's an interactivity. If you take this into the lab, so if you want to use this as an electronic notebook, you can. You can have checkpoints. You can tick off it once you've done each step, so you know where you've got to. You can have actually timers. I don't know if that's in this particular screenshot. We have timers within the step itself. I think actually you can see it down here. You can leave notes, and it's all recorded in a centralized place, and it helps you keep track of where you are. So it sort of helps you read and also actually do your experiment. Make notes of where things went wrong or whatever. It's also a Google Docs for lab protocols, like I said. So actually, you can work with Protocols.io cross-collaboratively. So I have my glamorous assistant, Tina, who is also hopefully in the protocol. And you can see I am typing over here. And in a minute, we should hopefully see her adding maybe a new step. Well, at least you can see, oh, there you go. So she's added step 5. She started working. You can see up here, she's working here. So we can work collaboratively, very much like Google Docs. And also, it's a GitHub for lab protocols. So what I mean by that is that it provides strict versioning and also forking of protocols. So this allows provenience tracking. So where was the sort of ancestry of the protocol? Where did it come from? Be able to track the exact changes that you make over time, and ideally why. Also by having forking, which is where you make big duplicates of a protocol, but made major changes. You can still track who actually originally wrote the original version, which you've updated. And because you get this versioning system, as you can see here, you can always point for each of your publications to the specific version of the protocol that you used. So you just have to make a small change, make that, publish that, get the DOI, cite that. But people can always go back to previous publications and find the older versions of this. And all of this on Protocols.io is publicly available and open access. So all the protocols are not behind paywalls. They're not stuck in a journal supplementary, which has been lost or deleted because the journal broke stuff. It's less likely that you're going to lose stuff because the publisher broke all of their links, as Springer Nature did last week. So it's accessible to everybody. People from the global south, who maybe libraries can't pay for the paywalls for Science or Nature or whatever, they can still get this critical information so they can actually replicate your work. Just a brief recap of what I mean by version control, because sometimes if you're not working heavily in GitHub or whatever, you might be familiar with that. What I mean this is by you write the first draft on whatever platform, whether it's GitHub or Word or something like this. You make minor edits. You can actually record sort of what the changes you have made. And then once it's ready for publication, you publish this. And this makes a locked version of and sort of permanently, infinitely preserved version of that protocol. So no one can ever go to this version, to the link, and ever modify this. That exists for perpetuity. But this doesn't mean that you can't update it in a way. You can then make minor edits afterwards. So more, and do this step by step. You can again record each change, and then you make another version. This second version incorporates all of the edits you've made here, and this is the final version that you made, and you get another DOI. So let's say in the second publication, you can cite this to indicate that for this publication, we use this slightly tweaked version. But the nice thing is that you do have this link back to the original version. And so if someone wants to understand what differences there are, you can actually compare the two. So Protocols.io gives you an interface to compare different versions of protocols and try and understand why that is. This helps you with debugging, for example, if you want to understand why a particular protocol failed for whatever reason between two versions, and so on. So the critical thing is that these versions, once you give it DUI, this is permanent. You cannot modify this again. Someone can't go break it. Someone can't go, Zoom bomb it or the equivalent of that on protocols.io. I don't know. Yeah. So I myself am very much a doer learner. So I have to do stuff by trial and error. So I'm going to try and do this live now. I'm going to do two things. I'm going to firstly try to show you how you can run protocols and then introduce you to writing protocols. Let me just take the time. Okay. So to run a protocol, what I mean is that you can take an existing protocol, like this one here, one of the ones that the Department of Archaeogenetics and EVA have made. And let's say you're in a lab, you have a computer or a laptop next to you, which most labs are moving towards now. As far as I know, you can press the run button and press run protocol. And you can see here, if I move all the stupid Zoom stuff out of the way, you've got the protocol here, and you've got the different steps with a sort of progress bar and timer. So as I go through this, I can, in the lab, basically do that particular step. I press done, or I should press done. I don't know what this is not working now. Or is this 1 second? There we go. Sorry, the Zoom bar was covering it as a hidden thing. Going back, but you can see that basically it's done with step 1, step 2. On the right-hand side, you see the progress bar is going forward. Then step 4. And then somewhere there is a timer. Here, for example, so let's say you're in the lab, you get to this step, you want to run a spin down and you need to spin it for 1 minute. So here, rather than having to have a separate like kitchen timer, you can press start here and it'll start running. You can also leave notes here, so record that in your sort of log as well. It's recorded also on the right-hand side and things like this. So it is very interactive, and it's a way of helping you actually assist you in the lab for tracking things over time. So that's what I mean here, and you can actually go back to these. It is recorded in your profile, and I think you can also export them somewhere once you finished it. I can't find it at the moment. Probably after the saving run to basically use it as an electronic notebook if you wish. So that's running a protocol. I'm going to stop that timer because when I practice this, it turned on halfway through, which is quite embarrassing. It will even make a noise to tell you it's completed. And then to write a protocol, which I'll spend a little bit more time on, this is the protocols.io sort of homepage. Once you've registered, you have different workspaces. So you have a private space, you have different workspaces for different groups, so you can actually organize these in different ways. You can have your protocols listed here. You can press new to create a new one. And this is your writing interface. So this is what we saw previously when Tina was also collaborating with me. But you have different ways of inserting different steps, different types of steps. So we have a normal step. Then you can add a new section. Let's say, and hopefully what you should see is they've also got color coding here. So automatically adds the colors. So again, it helps you guide you through and keeps you track of where you are because we're more visual learners. You can add sub-steps. So I don't know, I have to think back to when I was in the lab. Purification, assemble, tubes and filter. I can add new sub-steps. You can track this here, blah, whatever. You know, also have conditional cases. So if I make a new step here, so if maybe you have two options of how you can do something, you can basically make different options if you need to do things based on whatever. You have a step case, and you can add different options here. So basically saying, if you do something by a 2-split purification or a 4-split purification or 8-split purification, you can say how you need to calculate things there. And then you have these different tabs right here. As with basically Word or Google Drive and stuff, you can insert links, you can insert images, videos, other attachments, you can include files, code blocks even, and do basic formatting like bold, italics, and things like this. You can add tables. And then again, talking about interactivity and things like this and sort of more richer interface. On the right-hand side here, you can see all the components. So these are things like, let's say I want to add a timer. Run for, I hate Zoom. Resume, if I can find the timer, please some duration. And I can say run overnight. And then maybe run for, let's say, 1 minute. And then when you actually would take this and run it, well, firstly, it guides your eye to, oh, this is a critical bit of information, what you need to run. But also, you can run it interactively in the, in the play mode. You can add, I don't know, notes. So make sure to do this. You can also set it to collapse as default. So this means that, let's say, if you want it for a new student, you can have this uncollapsed, but if it's something that you're running lots of many times, or you're quite familiar with it, you can have it collapsed. I don't know, there's lots of things, lots of toys and things you can play with. It's really great. You can leave comments while working collaboratively, and so drafting the thing, etc., etc. Then you have the description tab. This is sort of the preamble to the whole protocol itself. So a short description, you can abstract who it is, you can add multiple authors. And nice thing about this actually is once this is published in DOI, you can link this with your AaRC ID, and actually this counts as scientific output. It's an alternative thing from traditional journals, but you actually track this over time, use that for CVs, for applying for jobs and so on. You can give the original. So once you publish it, or if it is derived, the protocol is derived from a previous thing, you can actually put the citations here, funders, all this linking stuff. So it is considered basically another scientific output that is valid in most cases. Additional guidelines, ethics, if you have to, have photos of bones or teeth, which may be, sensitive, you can hide them as well, or give the warnings. More practical references, and another really cool thing that I quite like, although they changed it recently and it's a bit slow, so we'll see how this goes, is that when you're making your materials list, you can make it easier for other people or yourself to order this from manufacturers. Protocols.io has a reagents database. So you can search, let's say, food turbo, and this is where it's going to take a bit of time, which is very slow for some reason, but essentially, oh, there it works. You can see here that it comes up with the particular object it is. It has a catalog number, so you can go to the manufacturer and order it as you would have to through ordering systems of the different manufacturers. And you also insert this into the steps as well. You can add your own reagents if they don't have it in their database. You can also maintain for your group, like all the stuff you actually already have there. It makes the search faster. So this is really cool and nice. So actually, if you click on the actual reagent, it goes to this, this database where we used it, but it has all the information that you maybe want to use. So it's a very rich interface. Then once you're ready to publish, which I'm not going to do here, but you can basically go share. Oh, no, not share. More, post a draft, reserve the DOI. Once you reserve the DOI, that is ready for when you publish, then you get this stamp and you can see the final protocol. If anyone wants to see a fully fledged protocol, please shout, and one of the maybe Q&A people can post in the chat already their examples of stuff they've done before. Okay, so that's the very, very, very brief demonstration. Like I said, generally I recommend people just go and try it yourself and get familiar. The platform, the layout is actually relatively familiar if you run stuff like Google Docs, Drives, or Excel even. The main thing is just online. So at the last bit of the talk, I'd like to just spend 5 or 6 minutes doing the frequently asked questions. So these are things that come up often when I try to convince people to actually adopt a platform like Protocols.io. So one common thing is, I've already got 5 projects to do. I need to write my thesis. I don't have time to write it up separately. The same protocol I've already written up. Well, my argument is that honestly, you're already wasting your time, and not just yours, but everyone else's, or your peers and colleagues, I should say. So already, probably you're having to rewrite the same method section for each publication, but I mean that rewriting it and over again. Different phrasing, match, whatever requirement the particular publisher or journal requires. So that's a waste of time. then because you maybe have the word limit, you can't put all the detail there, you're making them, the other researchers, your colleagues, your peers, to work out all the details, the critical information missing from each citation and so on. And you also, extension that often you'll force researchers to go down quantitatively. Like I said, I had that image from that application, bad citation chains, which also, it takes such a long time to, like, actually doing that citation chain I showed you of the 4 things took me 5-6 minutes for a single protocol to not actually get the information I needed. Because, you click on the thing, you have to go to Crossref, open on the publisher website, then you have to maybe find out, is it in the main text or is it in the supplementary? You have to download the supplementary. You get my point. And so by using version-controlled protocol systems like Reproducible.io, after this initial investment, you don't have to rewrite your methods section every time. You make a small tweak to the existing protocol, publish that one, and cite that DOI. Also, because the interface is actually designed for reading protocols, it's a recipe method, it's much easier for your eyes to be drawn to the place where to get the critical information. If you're looking for, okay, which reagent which polymerase they used, you don't have to basically scroll through multiple paragraphs or black and white text to find this information, reading the entire thing. You can go to the materials tab and you'll very quickly find it, particularly if it's structured. And also in the lab, it's a better interface for running. You don't have to convert it to an Excel, your own version of an Excel sheet if you're trying to adopt the existing protocol. It's just ultimately much smoother if you have a single source of truth that is actually designed for you, for people working in the lab, not lazy publishers who don't want to change their publishing system. Other people may ask, oh, but why do I have to learn a different platform? I already have to learn 20 different ones. The sad truth of this is you have to do that anyway. There's a new social media thing every year that everyone is forced to use. Your university will be forcing upon you various platforms, for teaching and marking things, or your university or institution will require you to go through a new portal for recording your time or actually publishing, whatever. You're going to have to anyway. What difference does one will make? The added benefit here is that this platform like this actually will make your life easier and is better in the long run. So, like I said before, traditional academic article structure is not fit for 21st science. It cannot fit complex experiments, complex analyses, complex materials lists into this crappy base prose. Something you don't, or very few people, I should say, actually take and run in the lab themselves. You may also argue, oh, why do I have to learn this platform, whereas my institution already pays a lot of money for this particular thing we use internally? That's fine, but you're going to have to rewrite your protocols anyway when you publish it. So the stuff that your institutions are paying for are often not exportable or shareable outside your institution. So it's basically a paywall. Some people may comment already that if they're poking around the website, you only get 2 free private protocols in Reproducible. This is slightly annoying, I agree. But on the other hand, my answer is so. Most labs, I would argue, particularly in ancient DNA, less so in proteomics, this is a newer field, but most people, you're not developing new protocols. It's nothing to be scooped upon. In most cases, you're just making some minor tweaks, either for your slight particular sample set, because maybe, it's less well preserved, or maybe just adapting it to the machines that you have in your lab. It's nothing to hide. Some people, or very many people, often say, oh, but James, I don't want to do that because I'm too shy of uploading my code, but also my protocols, because what if I've made a mistake? And it's like, it's okay to make mistakes. Science is meant to be self-correcting. You just make a new version and point people to the latest version. That is completely okay. It's fine to make mistakes. It's nothing to be shy about. If anything, it'll be much worse if you're not open about this, because if you have hidden something gone wrong for years and years and years, you've wasted so much other people's money, and you'll be too shy to tell them that they've wasted their money on, using the wrong reagent or whatever. However, on the other hand, if you are shy, you can just ask your institution to pay for an institutional license, so you can have more private protocols if you need. Something adopted here for a while by the Department of Genetics is just have the PI pay for a single user license, and you should all share the password. That's not so bad. Or another hack you can do is each person writes privately. Let's say you've got 4 people in your group, that's 8 private protocols, and then you just transfer it to the common organization or group workspace once it's ready. So there are ways around this if necessary. Some people get worried, but when the original publication do citations, no, because you should always cite the original version of the original publication of the citation. What you should also be doing is citing that plus the version or variant you use. This is what you do anyway. Normally, you're saying I ran, so in ancient DNA, the Mayer and Kirschy 2012 library construction protocol with the following modifications. In this case, you're just condensing with the following modifications from the horrible prose-based text to a nice platform, which you get the information better, as you can see here. And so actually, both people, everyone wins. The original authors get their citation, you get your citation on your protocol. Some people may ask, oh, what are some alternatives to this? Actually, at the moment, I've not found a good competitor. So there's openwetware.org, which is like a 90s era wiki page. It's a bit janky. It's very open. It's very public, but it's just text. There's no rich interface and so on. And there's a lot of stuff on there, and it's quite hard to find things. There's another thing called Protocol Exchange from Springer Nature, or really Springer Nature have also bought over Protocols.io recently. It is better. It is open access, but it's not a rich interface in the same way that Protocols.io is. It's not interactive as far as I'm aware. And actually, most other things, if you search alternatives to Protocols.io, are actually just traditional journals like bioprotocols, things like this. So at the moment, at least, there is no, in my opinion, a good alternative. Maybe if someone knows something, please say. I'll be curious to see. Yes, so Protocols.io is pretty good. I was quite happy with it, and my department was quite happy with it when I forced them to adopt it. Anyway, so in summary, save your own time. Don't rewrite your same method section over and over and over again. Tweak, make a new version. Save your colleagues time. You can write rich and detailed protocols with all the details for maximum reproducibility. You're not hiding any important detail, not behind a paywall, so on and so forth, and save your own time. So you're saving your time by doing all of these. You don't have to deal with emails or questions from other people, like you can't replicate your analysis, and so on. And also you can track your own, your progression over time, and help with debug and things like this. Okay, so that's basically my spiel, which is traditional publishing sucks. Use cool, fancy, shiny things with colors and buttons and stuff. It's much more fun, and it helps a lot of people. And so with that, to end the talk section of this webinar, I'd like to thank generally the Department of Archaeogenetics, MPI EVA. So like I said, I semi-forced them to basically convert most of our internal protocols to Protocols.io, but I think that went very successfully. We still get a lot of citations for these, surprisingly. Also the Microbiome Sciences, who allow me to do this both wet lab, but also dry lab. Also SPAAM, HAAM, PASTA, and AaRC. Like Lisa said, I sort of kickstarted the HAAM community itself after stealing the name. But it's really nice to see sort of the same concept being adopted by many different people. And I should acknowledge my funding, which is the Max Planck Society, the Werner Siemens Stiftung, and the Leibniz Archive. So thank you very much. Do you want to do a brief Q&A of just my stuff, or do you want to go straight into the roundtable?
I mean, I figured your stuff is kind of the same topic as the roundtable in general. Yes, so at